Hello, good morning everyone. Today we are going to learn about enzyme inhibitors. What are enzyme inhibitors? These are any chemicals that will slow the velocity of the reaction, or I have written it as any chemical that diminish the velocity of the reaction, or they will completely stop the activity of the enzyme. Such chemicals are called inhibitors. These chemicals can be poison, they can be antimetabolites, they can be antibodies, or they can be cyanides. Overall, they should slow the velocity as well, change the velocity, as well as they should stop the enzyme activity. Then it is called inhibitor. Enzyme inhibition are of two types: irreversible inhibition and reversible inhibition. Reversible inhibitions are of two types: competitive inhibition and non-competitive inhibition. So first let us learn what is meant by irreversible inhibition. Irreversible, name itself indicate, that means once the inhibitor inhibits the enzyme, its action is permanently, activity of the enzyme is permanently stopped. I have written it as permanent inhibition it is. What happens exactly, let us understand. In irreversible inhibition, inhibitor will bind to the active site of an enzyme by a strong bond. Look at here, red colored one is inhibitor and this is my enzyme, and I have shown a line to indicate that there is a strong bond formation. Usually covalent bond is considered a strong bond. That bond is formed between inhibitor and active site of an enzyme. As a result, once inhibitor attaches, detaching inhibitor is not possible. Therefore, enzyme activity stops permanently. Enzyme cannot produce any product because it cannot bind to substrate also. That is known as irreversible inhibition. That is one way. Another way is inhibitor, by forming strong bond, attaches with the enzyme, so that once inhibitor binds, enzyme will completely change its shape. Active site is a site which is not having fixed shape. When inhibitor binds, enzyme shape changes. If enzyme shape changes, the product cannot be formed because substrate cannot bind to it. That is irreversible inhibition. Example for irreversible inhibition is, it is used in terms of beneficial way, whereby an inhibitor like penicillin is used to inhibit an enzyme, transpeptidase enzyme. Transpeptidase enzyme is required for bacteria to synthesize bacterial wall. If you introduce penicillin, it will stop or inhibit the transpeptidase permanently. Therefore, bacterial wall is not synthesized. As a result, the bacterial infection can be prevented. That is the benefit of irreversible inhibition, or you can consider it as an example for irreversible inhibition. Now we will move on to reversible inhibition. What is reversible inhibition? It is a kind of inhibition where inhibitor binds to enzyme by forming weak bond. In irreversible it was strong bond, now it will form weak bond, usually hydrogen bond and all. Since it is forming weak bond, when inhibitor is attached, enzyme activity will be lower, or enzyme activity will be stopping, but this weak bond can be broken by some means. Hence you can detach the inhibitor. Therefore, once inhibitor is detached, enzyme regains its activity again. Hence it is reversible. Reversible meaning enzyme can regain its activity again. It is not permanently inhibited. What are the ways by which we can reverse the inhibition reaction? Say that to an enzyme, inhibitor has bound. Now how can I remove this inhibitor is increase the substrate concentration. When you increase the substrate concentration, substrate will knock off the inhibitor, will break the bond, knock off the inhibitor from the active site. Hence now enzyme is free of inhibitor. It can bind to substrate and produce product. So that's how we can reverse the inhibition. These reversible inhibitions can be of two types: competitive inhibition and non-competitive inhibition. Let us understand what is competitive inhibition. In competitive inhibition, this is my enzyme, this is the substrate of the enzyme. Look at the inhibitor. Inhibitor and substrate closely resemble each other in terms of structure. They look similar. Also here, substrate, it binds to active site. Inhibitor also, now because of similar shape, it will also bind to active site. So there will be a competition between substrate and the inhibitor. That's why it is called competitive inhibition. Hence, competitive inhibition is a type of inhibition where inhibitor resembles with the substrate. So therefore, they will compete with the substrate to bind to active site. That is competitive inhibition. What are the examples we have for competitive inhibition? Look at the example. Succinate is the original substrate of enzyme succinate dehydrogenase to get a product. But there is an inhibitor called malonate. It looks exactly similar to succinate. So malonate, what it will do is it will go and it will compete with the succinate to bind to succinate dehydrogenase. If malonate binds to succinate dehydrogenase, enzyme activity is stopped, inhibited. That is why it is competitive inhibition. Second example of competitive inhibition is it is a beneficial aspect. Competitive inhibition can be used to control pathogen. How? Pathogen like bacteria, it converts by using a precursor, it forms folic acid. Folic acid is one of the important bacterial component. To prepare this folic acid from precursor, bacteria needs PABA. Para-amino benzoic acid is required as a precursor. Now, how to control bacterial component synthesis? What you have to do is give sulfur drugs. Sulfur drugs is an inhibitor of PABA because sulfur drugs and PABA closely resemble each other. It will go and inhibit the sulfur drugs will inhibit the PABA. It will not resemble, instead inhibit the PABA. As a result, there will be no folic acid synthesis. That is the second example of competitive inhibition. Now, what will happen to the Vmax and Km of the reaction during competitive inhibition? Look at the graph. On the y-axis I have taken velocity of the reaction, x-axis I have taken substrate concentration. Normal graph, how it will be? As the substrate concentration increases, enzyme activity will also increase. Then enzyme activity reaches maximum point. In that maximum point, all the enzymes are saturated with the enzyme. Therefore, further if you increase substrate concentration, enzyme activity remains constant. So this is the Vmax point, whatever I have written in pink, Vmax. And if this is Vmax, this point indicates half of the maximum velocity, Vmax by 2. If you extend half of maximum velocity, it will touch the graph over here, then extend it downwards. Where it touches the x-axis, that point is called Km. What does Km indicate? Km indicates substrate concentration at which half of the maximum velocity is reached. It also indicates affinity of the enzyme, which means what is the affinity of an enzyme towards the substrate, that is indicated by Km, which in one more way to tell, Km tells us whether and how much that one particular enzyme loves the substrate. That is known as affinity. If in competitive inhibition, because of the presence of inhibitor, velocity of the reaction will be slower, velocity is less, therefore graph has come down. Slowly reaction will occur. It will reach Vmax somehow, because as you increase substrate concentration, inhibitor action can be prevented. Somehow Vmax is reached, but not at the same substrate concentration. Look at here, first graph has reached Vmax at this particular substrate concentration, whereas in the presence of competitive inhibitor, Vmax is reached at this particular substrate concentration. So Vmax in the competitive inhibition remains same. Now take the Vmax by 2. This is same point, Vmax by 2. But if you extend, you find that Km value has increased in the presence of competitive inhibition. So Km increases. What do you mean by Km increases? Affinity of a particular enzyme towards the substrate in the presence of competitive inhibitor decreases. I have written it as affinity decreases. So that is about competitive inhibition. Second type is non-competitive inhibition. In case of non-competitive inhibition, you know that enzymes will be having a pocket-like structure called active site to which its substrate binds. Now there is an inhibitor which doesn't resemble the substrate. If it is not resembling the substrate, it cannot bind to active site. Still the inhibitor chemical will inhibit the enzyme. How it will inhibit the enzyme is, in the enzyme, apart from active site, they have got one more pocket-like structure called allosteric site. Inhibitor goes and binds to the allosteric site. When inhibitor binds to allosteric site, enzyme activity gets stopped. That is one way of stopping the enzyme activity. One more way is, when inhibitor binds to allosteric site, there is a signal given from the allosteric site to active site. As a result, active site also changes its shape. When active site changes its shape, further substrate cannot bind, enzyme activity is stopped or velocity of the reaction diminishes. So that is non-competitive inhibition. Hence, how to define non-competitive inhibition? It is a type of enzyme inhibition whereby inhibitor do not resemble the substrate, they will not compete with the substrate. Instead, inhibitor will bind to the allosteric site and diminish the velocity of the reaction. Now let us look into what will happen for the Vmax and Km value. This is the normal graph. As the substrate concentration increases, enzyme activity increases, it reaches Vmax, remains constant then. This is the Km, blue colored one is Km for normal enzyme activity. Now in the presence of non-competitive inhibitor, velocity of the reaction when there is inhibitor decreases, it will be very slow. Vmax is not as before, it will be lesser than the before point. Therefore, Vmax in the non-competitive inhibition decreases. That is one point to note. When Vmax decreases, Vmax by half also comes down. If this is the maximum velocity, it's half of the maximum velocity has reached at this point. When you extend, you find that Km of non-competitive inhibitor is same, inhibition is same as that of before, which means in non-competitive inhibition Km remains the same and the affinity of the enzyme towards substrate remains same. It will not change, but the overall velocity of the reaction decreases. That is non-competitive inhibition. Example, cyanide is a non-competitive inhibitor. It will inhibit the respiratory enzyme cytochrome oxidase. So that is about enzyme inhibitor and their classification and the types of inhibitor. If you have liked it, please click the like button and subscribe. Thank you.